Swayam Prabha. Digital India. Educated India.
So, I will take on from where I left last time. I was talking about immunodeficiencies, under which I will go over, I had gone over very quickly over a couple of slides, and I will take some time now to go through them a little bit in detail. Now, immunodeficiencies of the immune system is very well known. You have immunodeficiencies of T cells, B cells, as well as complement components. Now, immunodeficiencies of the T cells will be dealt with in another class. I will restrict myself mostly to immunodeficiencies of B cells. Now, lymphoid immunodeficiencies, which would mean that any deficiencies which would lead to the development of the lymphoid series would affect, of course, both the lineages T cells as well as B cells, and this therefore would lead to severe combined immunodeficiency. SCID, in short for severe combined immunodeficiency, this can be caused by different genetic factors. However, all forms of SCID have very low numbers of lymphocytes, and even if there are lymphocytes, even in low numbers, it has been seen that the few circulating cells which you see, lymphocytes that you see, they do not respond to a antigenic stimulus, as has been studied by stimulating with mitogens. Therefore, now lack of T or B cells would result in severe recurrent infections. Now, these infections, unless such people who have these deformities are kept in isolation away from the regular atmosphere, this condition would lead to lethality, and that too, of course, in early years. Let us see the defects that can underlie SCID, severe combined immunodeficiency. The most common defect under SCID is mutation in the interleukin 2 receptor and the gamma chain, that too. Now, interleukin 2 receptor, I hope this rings a bell. Interleukin 2 receptor is absolutely essential for the signaling of T cells. Interleukin 2 receptor is also required for B cells, but you have also interleukin 4 and 5 receptors that can take over. Now, mutations in this receptor affects also other receptors such as interleukin 4, 7, 9 and 15, because the same gamma chain is associated, makes a part of these receptors. So, ultimately, therefore, one can imagine that mutations in IL-2 receptor can in fact bring about immunodeficiencies, severe immune deficiencies, because other receptors also get involved. RAG1 and 2, recombination activating genes 1 and 2, the products of which are required for the development of both T and B cells, because these enzymes, the products of RAG1 and 2 genes, enzymes are required for the T cell receptor organization at the gene level as well as immunoglobulin gene organization. So, defects in either RAG1 and 2, mutations in either RAG1 and 2 leads to severe combined immunodeficiency, and you might remember that I said there are mice that do not have RAG1 and 2, and these mice therefore lack lymphocytes and can be used as animal models for adoptive transfers of lymphocytes from other animals, as well as one can also test humans. In this, so RAG1 and 2 deficient mice are very good animal models for studying immune responses restricted to T and B cells. Now, there are also defects that mutations of ZAP70, and this is one of the common ones, and therefore, I am describing these to you. ZAP70 is the Syk kinase which is required for signaling. Now, deficiencies in this particular gene for ZAP70 would result in lack of signaling of normal lymphocytes. So, there are lymphocytes which develop normally, but because of lack in ZAP70, the signaling event is very much curtailed. Now, there are SAR kinases of course, but the importance of ZAP70 in fact or the psych kinases become very apparent because of the description of these deficiencies. So, there are normal lymphocytes, but signaling is affected, and therefore, of course, mounting an immune response to the optimum level is compromised in these individuals. Let us look at now other immunodeficiencies such as X-linked A gamma globulinemia. Now, this is also known as Bruton's hypogammaglobulinemia. Hypo, which would mean that there is mutation in the Bruton's tyrosine kinase. If you remember, when I described the signaling events that goes on in B cells, Bruton's tyrosine kinase is one, activation of which is one of the arms of the downstream processes from the ITAMs or the immunoglobulin receptor activation, which activates the ITAMs, activates therefore a large number of molecules, which is because of phosphorylation. Now, BTK, Bruton's tyrosine kinase, is again another phosphorylating enzyme, and defects in this has been shown to have severe effect on the development of the B cells themselves. And it has been seen that B cells under these conditions remain in the pre-B cell stage, though rearrangement of the heavy chain germline light chain gene has taken place, but these cells remain in that pre-B cell stage and cannot now go on with further development, and therefore cannot become mature cells, and therefore there is lack of these cells in circulation, mature cells in circulation. Obviously, such individuals come down with severe infections. Another condition is X-linked hyper IgM syndrome. I had briefly mentioned this when I talked about the signaling again, where T and B cells interact, and this interaction is necessary for both T cells as well as B cells. Now, in these individuals who have X-linked hyper IgM syndrome, as the name suggests, they have extremely high levels of IgM. Now, normally in circulation, the amount of IgM is about 1.5 milligrams per ml, but in these individuals it goes as high as 10 milligrams per ml. And there should not be much of a problem with respect to such high levels of IgM, but these individuals do not have IgG, IgA or IgE, any one of the isotypes subclass immunoglobulins. That is because there is no class switching seen in these individuals. If you do remember, of course, that IgM is the first antibody that is secreted in the primary immune response, and cells, B cells, memory cells undergo class switching from IgM to any one of the isotypes. For to secretory proteins, the class switching would be to IgG. Now, this requires the interaction between T cells, activated T cells, which express CD40 ligand, which is recognized or which binds to the CD40 receptor on B cells, and this is what affects or gives the competence signal to B cells. Now, if there is no CD40 receptor interaction, then there is no isotype switching. In fact, there is no memory cell generation, no isotype switching, and therefore, these individuals would make only IgM. Now, what should be the problem with just IgM? Along with the isotype switching, if you remember, there is also affinity maturation. If you might remember, the affinity maturation is because of error prone polymerase, and this happens only because of antigen mediated stimulation of memory cells. So, if there are no memory cells, therefore, no isotype switching, and therefore, only IgM. Because there are no memory cells, these individuals also do not have germinal centers. So, they have these are highly immunocompromised situations. Selective deficiencies of immunoglobulin classes have also been seen. There are individuals who do not make IgA or E or Ig, A, E and G. The most common one under these deficiencies is lack of IgA. What we talked about earlier is lack of any of the classes, but you can have selective deficiencies. Now, lack of IgA could be because of mutations in the switch region, which does not allow switching of IgM to IgA. Such individuals have recurrent respiratory and genitourinary tract infections, and one can imagine why these type respiratory and genitourinary tract, because you do remember that IgA is the class of immunoglobulin or the antibody which is required, which in fact is present in the lining of the mucosa. So, lack of, and that is where IgA is extremely efficient with respect to counteracting pathogens. So, there are recurrent such infections. Genetic deficiencies of the complement components, I have also mentioned this to you earlier when I taught you the complement cascade, that there are homozygous deficiencies of early components of the classical pathway known C1q, C1r, C1s, C4, C2. Now, lack of any one of these or mutations in any one of these definitely would lead to abrogation of the classical complement pathway. Such individuals again would have recurrent, not only recurrent infections, but also they exhibit marked increase in immune complex diseases. That is because the complement component C3 is extremely efficient in the process of opsonization, also extremely efficient in removal of complexes by the receptors, through the receptors present on phagocytes. Now, if there is no triggering of the complement cascade, the split products are not formed, and therefore, there would not be any clearing of the immune complexes. So, increase in immune complex diseases such as SLE as well as glomerulonephritis. Now, these are secondary as opposed to what we talked about with respect to the autoimmune diseases. In respect of complement components or deficiencies, these individuals who have C3 deficiencies have the most severe clinical manifestation because of the reasons I just mentioned. Also, apart from the classical components, individuals with deficiencies either in factor D and propidin have also been reported, and these are associated with specific infections, Neisseria, but these individuals do not have the immune complex disease. One can understand why, because the immune complex disease is because of lack of C3, which is absolutely required for the clearance of immune complexes. Now, as I said earlier, there are of course equal number of immunodeficiency of the T cells, but that will be dealt with in a separate lecture. Now, I come to the subject of cancer and the immune system. Why does cancer come under immunodeficiencies? Why do we study this disease, cancer, under the immune system? Now, it was, I will just like to tell you a little bit about the history of cancer and the immune system. Now, this concept, there is a concept of immunosurveillance. It was as early as 1909 that Paul Ehrlich proposed that the immune system prevents the outgrowth of carcinoma that would otherwise occur with high frequency. And there was another study that was made not as early as 1909, but somewhere around the 1950s and 60s, where it was seen that all individuals, individuals who die of old age, die not of cancer, but with cancer. What would that mean? That mutation. Now, how do cancers arise? That is, everybody knows, this is because of accumulation of mutations. Now, it was seen that all of us are exposed to carcinogens, those chemicals which can, or physical agents that can induce cancers. Now, how do these induce cancers? Is because of mutations. Now, every time there are mutations that are taking place in our cells, any molecules, we have an extremely immune, a sensitive and robust immune system which looks at these cancers, looks at these cells which now have modified, and therefore, these cells get cleared. In case, it has been seen that in the immune system also starts to degenerate, and it is related to age, with age the immune system starts to degenerate and therefore, accumulated mutations slip by. Therefore, now the cancers would arise. So, in old individuals there may be cancers that have established themselves, but before the cancer has taken its toll, the individual dies because of other reasons, maybe because of the circulatory system failure or kidney failure or whatever. So, Paul Ehrlich said that immune system prevents the outgrowth of carcinoma that would otherwise occur with high frequency. Soon after that, McFarlane Burnett proposed that tumor specific neoantigens were capable of eliciting a protective immune response. So, these actually go hand in hand. So, Burnett described these neoantigens. Now, how is the immune system and this immunosurveillance for that matter? Is there any evidence that the immune system must be in place to reject newer cancers which might be developing, or the immune system has a role to play in regression of tumors? The physicians have, there are a large number of reports in literature, physicians have recognized rare but spontaneous regressions and remissions in cancer patients. One such cancer, that of Burkitt's lymphoma, which is associated with Epstein-Barr virus, in these individuals there are often spontaneous regressions, and this can be associated with to a great extent by circulating antibodies, but more so by activated T cells against cancers, the cancers which are Burkitt's lymphoma. So, therefore, the immune system plays a role in allowing the cancer or not being able to mount an active fight against the cancer. Also, evidence for the immune system being responsible for not allowing or allowing cancer to develop is in partial tumor regression. Individuals who have had tumor, now it regresses spontaneously. Histologic evaluation of such tumors has been shown to have in large number of infiltrating lymphocytes or leukocytes. Now, this would mean that the immune system in fact is active in bringing about tumor regression. There are therefore, now with all this in the background, there is now an effort to develop numerous therapeutic agents which are designed to modulate the endogenous immune response in order to treat tumors. We will come to that in the next lecture, but let us look at what has already been established to some extent. Now, this of course is not the mode of treatment now, but the range of these agents to modulate the endogenous immune response is from injecting Mycobacterium bacillus, BCG, to treat bladder cancer. Now, what is the reason for this is BCG can activate macrophages, activate the immune system, activate leukocytes. Now, bringing therefore these cells to the site of the cancer can be beneficiary to the regression of cancer or treatment of cancer. Also, now to the other end of the spectrum, this is the simplest. The other end of the spectrum is to activate the immune system in a manner by inhibiting negative regulators. You may have studied by now the molecule CTLA4, which is a negative regulator of cytotoxic T cells. Now, if CTLA4 is inhibited, then by let us say blocking antibodies, then there would be no negative regulation and therefore, there would be enhanced cytotoxic T lymphocytes to the cancer cells. Let us go to the ultimate, to be able to understand the immune system with regard to cancer or cancer and the immune system. So, first we need to understand what is cancer and how does it develop. So, what is cancer? The definition is very simple. It is uncontrolled proliferation of cells. uncontrolled proliferation, which would mean of course there should be some kind of a regulatory mechanism which allows cells to renew and die. This system, this process is known as homeostasis. Cells are short lived. Of course, depending on the type of lineage or the lineage, cells can live for 3 to 4 weeks, and some cells like RBCs would live for a few days. There is a constant need for cell renewal. Whenever there is cell death, there is cell renewal, and this is an extremely tightly regulated process. However, in certain conditions, you can have uncontrolled proliferation of cells once they have started to proliferate. So, there is now, one can explain this in a little bit easier way, that if you take away a small slice of a liver from an animal, okay, now there would be intense proliferation of the cells where the cells have been destroyed, and this proliferation goes on at a heightened pace until that little portion has regained its structure. This happens, we are constantly being inflicted with small cuts and bruises, and you will see that the skin finally comes to its normal self. There is no outgrowth there. There is no more cell division after attaining that particular contour which was there before. However, in case of tumors, there is this control or regulation is lost. You can have, this mass of cell is called a tumor. The tumor can be either benign or malignant. I think this is something which is quite well known, and the benign tumor of course is a mass of cell which is restricted. It is in fact isolated and compartmentalized. Benign cells therefore, benign tumors therefore can be treated very easily. Malignant on the other part is when cells now grow out of this mass and go into circulation and can now establish themselves elsewhere. Let us look at how this can happen. I would like you to remember that usually tumor cells arise from one single cell. One can call all cancers monoclonal. All cancers, though later on might be a huge mass of tumor, would arise from a single cell. Of course, from a single cell, when it becomes a huge tumor, there would be of course large number of processes by which that single cell has changed. So, its progeny has changed to, well, we will talk about this later, because that is one of the mechanisms by which cancer cells can evade the immune system. But suffice here to say that a tumor cell arises from the tumor arises from one single cell. So, this is an aberrant. Now, this is an epithelium, you can see underlying there are blood vessels, and this is the stroma. Now, a single cell which may be because of development of accumulation of mutations. Now, this has established itself as a tumor cell. The tumor cell starts to proliferate and become a mass of cells. It then, if it is a malignant tumor, then it starts to invade, go outside of this epithelial compartment, starts to go into the stroma. Not only that, these cells can also enter the blood vessel. That is what is shown over here, that the tumor cell now starts to metastasize, enters the blood cells, and now through the blood it can go elsewhere. Metastatic tumors are cancers, and those are the ones that need of course treatment. Malignant tumors are classified under 3 categories: carcinoma, leukemias and lymphomas, and sarcomas. Carcinomas, now this is dependent on the lineage of the cells, the lineages from which they arise. For example, carcinomas are tumors of endodermal or ectodermal origin. Leukemias on the other hand are tumors of the hematopoietic lineage. Sarcomas are tumors of the mesodermal origin. Now, carcinoma account for 80% of the cancers known today. Examples: colon, breast, prostate and lung cancers. Leukemias and lymphomas. Now, leukemias are single cell cancers which are formed as single cells, whereas lymphomas as tumor masses. Sarcomas are tumors of the bone, fat and cartilage, and they are quite rare. Now, I said carcinomas form 80% of all tumors, leukemias and lymphomas are about 10 to 12%, and sarcomas are much lower. Now, there must be something, some agents which we know that these are carcinogens, cancer causing agents, which actually are responsible for inducing mutations. Chemical agents which could be DNA alkylating, that means DNA modifying agents, or physical agents such as ultraviolet light, ionizing radiation. Now, these are carcinogens. The role of mutagenesis, I have been saying all this while that cancer arises from accumulation of mutations. Now, how was that established? That mutations in fact is what cancers which leads to cancer development. One very good example, now there are several such examples. The one that I can immediately tell you is a condition known as xeroderma pigmentosa or skin cancer. Now, in individuals with this particular condition lack a particular DNA repair enzyme. Now, apart from the DNA repair that I have talked about with respect to immunoglobulin gene organization, I am sure you can appreciate that DNA repair enzymes are absolutely required even during the DNA, during transcription stages. DNA is prone to get damaged by several mechanisms, and there are DNA repair enzymes. Individuals who lack these repair enzymes, especially the one which is specific with respect to the ultraviolet, UV specific endonuclease. This repair enzyme is required. Individuals who are exposed, who have this condition, who lack this DNA repair enzyme, when they are exposed to UV, in fact, just being in the sun for a long period of time, these individuals develop skin cancer. So, this is direct evidence of mutation and cancer. There are viruses which are also known to cause cancer. They are also they are not chemical reagents, but they are also cancer causing agents. Examples are HTLV, human T lymphocyte virus, HPV. Now, HTLV is associated with T cell leukemias, HPV cervical cancer, and HPV of course has been in the news in the last couple of years, because there are vaccines available now which can control cervical cancers. HPV liver cancer, EBV Burkitt's lymphoma as well as nasopharyngeal carcinoma. So, these are viruses which are known to be associated with human leukemias and cancers. Now, how do viruses in any way, how are they associated with cancers? Now, viruses are known to have oncogenes. Onco is the name for cancer. So, genes which are associated with cancer. For example, I will talk about this a little later, but oncogenes, for example, VSARC from Rous sarcoma virus. Now, this of course, Rous sarcoma virus is a virus in mice. The VSARC, when this particular gene of the Rous sarcoma cloned and then transfected into normal cells, establishes malignancy. So, again directly relevant oncogenes which have been shown to cause cancer by simple transfection or introducing this gene in normal cells. Now, let us talk about oncogenes and cancer induction in respect of other cancers other than Rous sarcoma virus. How early? It was 1971 that Teminch suggested that oncogenes are acquired by the viruses from cells. So, those oncogenes that are the VSARC that I talked about should have its partner in normal cells. What therefore Temin said that these viruses, when they infect, they have picked up these genes from normal cells, and he called them proto-oncogenes. That means oncogenes which are in the form where they are not oncogenes, or the initiation of oncogenes, proto-oncogenes. Bishop and Varmus later identified genes called C-Sark in chicken cells, normal chicken cells, and these he showed were homologous to the V-Sark in Rous sarcoma virus, RSV Rous sarcoma virus. Later on, more and more oncogenes were studied, and it was recognized that the cellular oncogenes and the viral oncogenes are highly conserved. The cellular oncogenes, as one can imagine, have introns, whereas viral oncogenes do not. We know therefore that viruses have acquired the C-onc from cellular RNA and not from the DNA, because the C-onc have introns. That means cellular, normal cells have these genes and there are introns. So, these viruses, where have they picked up? They have picked up after these genes have been transcribed, and therefore, they have taken from cellular RNA, the messenger RNA. Cancer associated genes, what do? Do they have functions? Yes, they do have normal functions in the cells which are absolutely essential, that can be categorized under 3 groups. 1, induction of cellular proliferation, then inhibition of cellular proliferation. Now, why are we talking about induction and inhibition? Remember that I said homeostasis is an extremely regulated process. You have proliferation taking place as soon as there is cell death, but this proliferation, cellular proliferation, is stopped as soon as now adequate numbers are formed. So, therefore, lack of any either of these can lead to abnormality, and we will see how. Thirdly, regulation of programmed cell death. These are the 3 cancer associated genes are associated with 3 normal functions. Let us first come to induction of cellular proliferation. Now, one type of oncogenes encodes protein that induce cellular proliferation. Very well known are growth factors or growth factor receptors. Secondly, now, before we come to that, so how do these cancer, how are these proto-oncogenes, how are they associated with cancer? Is if there is mutations, and therefore, when the growth factors should be X numbers, and they now, because of these mutations, are the expression is heightened, then either the growth factors or the receptors being expressed in an appropriate way would now lead to uncontrolled proliferation. The oncogenes which are associated with this are NU and ERB, and there are several others. I have just taken 2 examples. NU is related to EGFR, epidermal growth factor receptor, and ERB is the receptor for epidermal growth factor. Second type of proto-oncogenes encode proteins that function in signaling, and or they could be transcription factors. SARC, again this is something that you remember, are proteins which are absolutely required for signaling in B cells and T cells. So, SARC and Abelson, these oncogenes encode tyrosine kinases which are required for signaling. RAS is a GTP binding protein. MYC, JUN, FOS, these genes encode transcription factors. So, now these all are required in the normal function of growth and signaling and growth. If there are mutations therefore in any of these, this can lead to uncontrolled proliferation. Now, these are genes which are associated with proliferation either at the level of growth factor themselves, the receptor and the ligand itself which induces signaling. So, there plus signaling molecules which now are mutated in a manner that there is no control, can lead to extensive proliferation or uncontrolled proliferation. There are proteins which inhibit, there are tumor suppressor genes. Inhibition of cellular proliferation is the next. There are tumor suppressor genes. These suppressor genes encode proteins that inhibit excessive proliferation. So, therefore, what we know as tumor suppressor genes in fact are the ones that are playing a role in the normal process of proliferation. One such gene is retinoblastoma, the other one is the very well known P53. Inactivation or mutations in any one of these would now lose, such cells would lose control on the control of cellular proliferation, unchecked proliferation. Therefore, the mutations of these two genes are involved in RB is involved in the establishment of retinoblastoma, and therefore, in fact, the gene was named RB because it was first isolated from retinoblastoma. P53 mutations have been reported in more than 90% of small cell lung carcinoma and more than 50% of breast and colon cancer, and of course, in several other carcinomas. So, we have seen mutations of those genes, the products of which induce proliferation, growth factors, and those that are induced are involved in signaling, which ultimately again go on to cell proliferation, or the proteins that can check proliferation. Mutations of any of these can lead to uncontrolled proliferation. There are proteins which are also a large number of them in fact, which regulate programmed cell death. Now, programmed cell death, this is or apoptosis. Apoptosis is programmed cell death. Apoptosis is a phenomenon which is absolutely required right from the development, from the embryonic stage where differentiation is taking place up to in adult life. Cells, especially one can think of cells which are undergoing mutations. There are proteins which can guard these mutations. In fact, I mean in a way that the proteins, when they are able to recognize mutations and therefore stall the cells in a particular cell cycle. There are pro-survival proteins and there are anti-survival proteins. Those that are pro-survival proteins such as BCL, anti-apoptosis protein, that means pro-survival, something like BCL2. Mutations in these again would not allow a cell to undergo apoptosis, and therefore, there would be uncontrolled proliferation. Going back now to the same thing that I said, proto-oncogenes can be converted to oncogenes and mutations in the proto-oncogenes by carcinogens. So, this is where we link. We know that carcinogens are cancer causing agents. They are the ones, they bring about mutations in the proto-oncogenes, making them oncogenes. It has been shown that single point mutation in the CRAS, another oncogene, is seen in several cancers, and chromosomal, the next one is chromosomal translocation. Movement of a proto-oncogene from one chromosome to another can also lead to cancers. I will talk to you about this translocation in a while, before which I have to introduce to you tumors of the immune system. Tumors of the immune system. Lymphomas proliferate as solid tumors. I have already mentioned that to you with a lymphoid tissue, and these are Hodgkin's and non-Hodgkin's lymphoma. Leukemias proliferate as single cells. Their numbers increase in the blood, whereas in case of lymphomas, they stay on as solid tumors in lymphoid tissue. Well, not really solid in the way carcinomas are, but they are enclosed in lymphoid tissue, whereas leukemias, which proliferate as single cells, they circulate in the blood. Leukemias could be acute or chronic. Acute are cells which are less mature in the development of T and B cells, and I have dealt with you will have acute cells or chronic. Now, chronic would be mature cells. If you are talking about B cells, those would be the ones which already have IgM and IgD on the cell surface. Acute would be a step before, pro or pre B cell stage. So, acute leukemias are known as acute lymphocytic leukemia or acute myeloid leukemia, depending on which lineage. Chronic would be chronic lymphocytic leukemia or chronic myeloid leukemia. Now, I will come to the translocation. A number of B and T cell leukemias and lymphomas have proto-oncogene translocated in immunoglobulin genes or the TCR genes. Very well known is the Burkitt's lymphoma, where the c-MYC from chromosome 8 is translocated to the immunoglobulin gene cluster, which is present on chromosome 14. Now, how does this happen? I mean, how does this lead to cancer? You can see, if you remember the immunoglobulin gene organization, this is the heavy chain, and this immunoglobulin heavy chain has already undergone recombination, giving VDJ. Now, if you remember the immunoglobulin gene organization, you have the promoter, which is on the 5 prime site of the leader sequence, which is on the 5 prime site of the VDJ. You have the enhancer sequence on the 5 prime site of the switch region genes. Now, there are different conditions, 2 of which are being depicted here. You have the c-MYC exons, c-MYC which comes from chromosome 8, it is translocated just before the enhancer gene, and therefore, now you have extremely high expression of c-MYC. Now, there are of course, these are translocation of c-MYC on this with respect to gene 1 and gene 2. Tumor immunology. What is tumor immunology? The study of antigens on tumor cells and the immune response generated to these tumor antigens. So, are there any tumor antigens? That is why in fact, we know why we are studying cancer under immune system is because I said that any mutations that take place in cells may bring about change in the cell surface expression of certain molecules, and people have seen that there is an immune response that can be generated to these antigens. So, are there tumor specific antigens? Yes, tumor antigens can be studied, or tumor specific antigens are known to be either tumor specific transplantation antigens or tumor associated transplantation antigen. Now, I will describe what each one is, but I would like to also tell you that several years ago, when inbred strains of mice were not available, cancer biologists were doing experiments where they transplanted tumors from one mouse to another and found that almost always tumor regression did take place. It was only later they found that that was not because of tumor specific antigens, but the regression was because of graft versus host reaction, because of the expression of class 1 molecules on nucleated cells. So, they had described these as transplantation antigens because all the experiments were done from one animal to another after transplanting these tumors. So, tumor specific transplantation antigens occur on tumor cells and not on normal cells, and this is because these are of course tumor specific. So, they occur on tumor cells because of mutations. On the other hand, tumor associated transplantation antigens are expressed on cells during fetal development, but these get reactivated during cancer development. So, they are tumor associated. We will deal with this in a little while. Were there any experiments that were carried out to show that tumor specific antigens in fact can induce an immune response and whether this immune response is protective? There are classical experiments, a large number of experiments. I will just tell you this experiment, which is a prototype experiment. Tumor specific antigens can have been seen to be expressed in tumors which have been established by the carcinogen methylcholanthrin. In fact, this is one chemical carcinogen that is utilized extensively, or ultraviolet light induced tumor cells. So, mice that are now which have received methylcholanthrin develop tumors. Almost 80% of mice that receive this with this reagent have been shown to induce tumors. Now, if these tumors are, I'll describe the experiment. The tumors is now resected and single cell suspension made, and these can grow as cell lines. Now, if such cells are irradiated so that they cannot multiply anymore, so cannot establish a tumor, and such cells when injected into a syngeneic mouse, which is of the same haplotype, the same HLA molecules or MHC molecule in case of mice, so that the rejection will not be because of the MHC class 1 molecules. So, now these a healthy mouse when injected with these irradiated tumor cells, a small number anyway, even let us say 10 million, subsequently a few days later, when the same mouse which has received these irradiated cells is injected with a lethal number of 100 million cells, survives. And this survived when the immune cells were then analyzed. This animal had T cells which could respond to the cancer cells effectively, the cytotoxic T cells, as well as had antibodies that could specifically bind to these cells, also induce or activate the complement. Now, an animal, a healthy animal which has not received these irradiated cells earlier, this animal would die. Now, this experiment has been done several times with several different cancers, and yes, showed conclusively that cancer cells have the capacity to induce an immune response that can be effective and protective. Virus associated tumor specific antigens, what I talked about is the one that is associated with carcinogens. Now, virally induced tumors express antigens which are of virus origin that are shared between all tumors induced by the same virus. Now, this is different from what I discussed earlier with carcinogens such as methylcholanthrin, where every tumor could be different. In case of virus associated tumor cells, all one virus, when it induces a particular tumor, most of the antigen expressed on this tumor cells would share antigens from between other tumor cells, but induced by the same virus. The examples are polyomavirus in case of mice, EBNA or Epstein-Barr virus associated nuclear antigen, HPV E6 and E7 in case of human. And I would like to just remind you that HPV vaccine in fact has been in the market for controlling cervical cancer, which is associated with HPV. Tumor associated transplantation antigens. We talked about tumor specific antigens so far. Now, we are talking about tumor associated transplantation antigens. These are not specific to tumors, they are present also in normal cells. Some of these antigens are expressed in low levels in normal cells, but at very high levels on tumor cells. Examples are growth factors and growth factor receptors. For example, transferrin growth factor, which is known as P97. There are about 8,000 molecules on normal cells, but these become 50,000 to 500,000 on melanoma cells. So, you can see that this means overexpression of growth factors. Now, why do cancer cells express these growth factors? Now, an example of this transferrin growth factor, what does transferrin growth factor do? It picks up iron, and therefore, increase in the number of receptors would make such cells very efficient in sequestering iron. Similar would be in case of other growth factors like epidermal growth factors. Increase in the number would now make the cell give the cell an advantage of picking up even small number of the ligands. Normal cells would of course would require many more molecules to be able to effectively pick up those that are required for their metabolism. Now, in association with talking about this P97 again, Now, recombinant vaccinia virus, which carries P97 gene, a recombinant virus, when injected into mice, protected them from challenge with P97 melanoma cells, again showing that one can generate an immune response to this P97 and this could be protective. Another set of oncofetal tumor associated antigens are AFP or alpha fetoprotein and carcinoembryonic antigen. These are what they call oncofetal because these antigens are expressed in the embryonic stage before immunocompetence has been established. Therefore, they are recognized. So, that means in the uterine of course condition when the embryo is expressing, then these would be recognized as non-self if they are expressed in adulthood. So, there are some cancers, especially now let us talk about alpha fetoprotein is present in alpha fetoprotein is present in milligram levels in the fetus. Now, this is very similar to the serum albumin, but the same protein is present only in nanogram levels in adult circulation. So, this is like a million times lower concentration in adult as compared to the fetus. In certain cancers, in liver cancers, alpha fetoprotein is highly elevated. Therefore, in fact, alpha fetoprotein is a marker, a biomarker for liver cancer. Where would it be used? That if the primary cancer is of liver, but metastasis occurs elsewhere, and in fact, the primary cancer is very small and has not been detected, then if alpha fetoprotein is measured or detected in the circulation of these individuals, this points to the person having a metastatic liver cancer. The other protein is carcinoembryonic antigen. It is a membrane glycoprotein on fetal gastrointestinal cells as well as liver cells. Now, this is CEA, this antigen is present in more than 90% late stage and about 50% of early stage colorectal cancer patients. So, this is also another biomarker. So, these proteins are present in the fetus and are down regulated in adulthood, but certain cancers express these proteins and therefore can be used as biomarkers. So, therefore, these are new antigens, though they are present, they are only present in the fetus and therefore, they are antigens of the cancer. So, let me just end here by I just like to go to the summary now, the last here before we come to the In summary, all cancers arise from accumulation of mutations that we have seen. There's enough ample experimental proof that is the mutations. In fact, what people have shown, they have been able to establish cancers in vitro in experiments by transfecting genes which are known as oncogenes, and also by creating inducing mutations. Now, cancers therefore arise from accumulation of mutations. Now, these mutations, all of us are so exposed to various all types of mutations, especially in the environment. We have so many carcinogens which are present. You all must have heard of carcinogens which come from the emission of vehicles. So, therefore, there is no way that we can avoid being exposed to carcinogens, but thankfully our immune system is so robust that the immune system can take care of these mutations, because the mutations that take place in normal cells by these carcinogens bring about expression of proteins on the cell surface of these cells which can be recognized as non-self. You may have by now studied natural killer cells which can recognize altered cells. Natural killer cells, if you remember, can mount an extremely ineffective immune response to the virally altered cells as well as cancers. If you think in terms of the innate immune response with respect to cancer, in fact, natural killer cells come to the forefront. So, natural killer cells should be able to eliminate any mutated cell which can be recognized as non-self. Now, if I told you that cancer arises, it's monoclonal. So, one type of cancer is from one type of cell, one cell. So, before the cell becomes a mass of cells, immune system, if it is robust, is able to mount an immune response, then these cells are deleted. In fact, this is what is happening to us all the time. Cancers of course, are detected effectively and thrown out of the system. However, some mutated cells can be detected and eradicated. Some mutated cells can persist because they go undetected. So, let us now understand this mechanism. A person who is immunocompromised may allow the tumor cells to go undetected. In an immunocompromised situation, a tumor cells has a better chance of slipping by, and once it slips, it is able to establish itself and the tumor becomes an entity. We do know therefore that an effective immune system or robust immune system is able to recognize cancers as non-self and is able to abrogate or delete this. However, we also know from whatever we've seen so far that most cancers are able to express new molecules on the cell surface which can effectively mount an immune response, curtailing the cancers. Now, in the next class, I will continue with this particular topic and tell you how cancers establish themselves. What is this mechanism by which the cancer is able to evade the immune system? In fact, there is not just one mechanism, there are several, and establish itself such that even if the immune system is robust, the cancer is free to roam around and establish metastatic foci in the rest of the body. So, I will see you next time with the topic on immune system and cancer. Continue with it. Thank you.